Okay, so we are going to continue with the pattern formation in early Drosophila, and we are going to focus on dorsal ventral polarity or axis formation, dorsal ventral axis formation, which we also know as DV axis, dorsal ventral. As compared to anterior posterior body axis formation, so different maternal genes are involved in DV axis formation or pattern formation. And just like in anterior posterior axis formation, here also we have very localized maternal proteins which are already present in the egg vitelline membrane or envelope. And at the ventral end, there is a ligand which is present for the receptor, and that ligand is very, very specific for the receptor which is here in the ventral membrane. Here it is shown. So you have on the ventral most side, which is this region, you can see. Okay, so if we draw, this is if we simply half the embryo and draw it on x-axis. So this is the dorsal and this is the ventral. So this is how we have drawn. The ligand is called Spätzle. It's very specifically cleaved only at the ventral side in this region. And binding of this ligand to the receptor activates toll signaling, which results in degradation of a protein called cactus. Cactus actually holds another protein, a transcription factor, which is called dorsal. So Spätzle actually leads to activation of cell signaling, which leads to degradation of cactus, and then dorsal moves inside the nucleus. If you pay attention here, in these nuclei, the nuclei are looking red, and these nuclei are white. So the red color is still in the cytoplasm. But here, the red color, which is representing the dorsal, has moved inside the nucleus. And this is showing you the concentration gradient of the dorsal. So only in the ventral most region, you have this movement. As I told you already, so the cleavage of Spätzle is very localized, and it's due to another small set of maternal genes, and one of the key genes is called PIPE, which is a protease, a heparon sulfate sulfotransferase. And this protease activity results in cleavage of Spätzle, a very localized cleavage of Spätzle. Spätzle binds to the toll receptor, which is already on the vitelline envelope, and then results in, and toll mRNA is also deposited in the oocyte. As soon as fertilization takes place, it gets translated, and then you have this receptor into the vitelline envelope. Although toll receptor is present throughout the membrane of the fertilized egg, but it's only active in the ventral side because of localized presence of Spätzle. And when you have active toll, you have movement of dorsal into nearby nuclei from the cytoplasm. This cartoon, alhamdulillah. So this cartoon shows you have dorsal held by cactus, in the absence of activation of toll signaling. As soon as you have Spätzle bound to the toll receptor, through palate and tube, signal transduction takes place, which leads to degradation of cactus and movement of dorsal into the nucleus. Okay, which is all this thing which we just talked about. Now, if you have a mutation in toll, if you have delta toll, we call such embryos dorsalized embryos, because dorsal will not move into the nucleus, and all the ventral part, so this is the dorsal, this is the ventral, all this region will become dorsalized, because dorsal will not move into the nucleus. All the nuclei will look like these dorsal nuclei. So that's why we call it dorsalized. Okay. If you simply transfer the wild type cytoplasm into tall mutant, it generates new dorsal ventral axis. Okay. So just like we were transferring the cytoplasm carrying bicoid, if you transfer tall, you can have new dorsal ventral axis here. And if you have active toll, we already know it degrades cactus, moves dorsal into the nucleus. And if you have delta cactus, if you have a mutant cactus, we call such phenotype the ventralized phenotype, because now dorsal will move everywhere inside the nucleus, even in the dorsal cells where cactus was holding dorsal in the cytoplasm. In the absence of cactus, now the dorsal will move into the nucleus, and all the nuclei from dorsal to ventral, ventral to dorsal, they all will look like ventral nuclei where dorsal normally moves into the nucleus. So we call such a phenotype ventralized. There are multiple functions of the toll. Toll is also involved in disease resistance, defense against different diseases, but early development, very early development, this crucial role of toll is extremely important, which is different than the disease resistance gene, disease resistance function of toll. Now, how this dorsal ventral axis formation takes place. As we just learned, dorsal is the key protein. It's if you have misregulation of dorsal, you don't see any ventral axis formation. And if there's no ventral axis formation, it means dorsal ventral pattern is disturbed. Now, how dorsal achieves all this? There is a cascade of gene expression which is activated. Dorsal is a transcription factor. As soon as it moves into the nucleus, it activates two genes. One is called twist and the other is called snail. So from here, when we go from ventralmost region towards dorsal, you can see there is a concentration gradient of dorsal. So very high to low concentration. The highest concentration of dorsal is known to activate twist and snail. And twist and snail, they suppress, they are also DNA binding proteins. They then suppress genes which are actually active in this region, which is called neuroectoderm. So movement of dorsal within the nuclei, the highest concentration of dorsal, this leads to mesoderm formation. So these green ones are mesodermal cells. These ones are called, on both sides, you see there's symmetry. This is neuroectoderm. This is part of ectoderm. And this yellow one is dorsal ectoderm. Okay. Now, due to gradient of dorsal, there are nearly 30 genes which are affected. And all this is happening, movement of dorsal within the nuclei. All this is still happening when cells are in syncytial blastoderm stage. They are not yet cellularized. Okay. So they are all these nuclei, and these nuclei are all sharing transcription factors which are present in the cytoplasm. Now, dorsal targets 30 genes, and it activates some of them as well as it represses some of them in the ventral region, which is this one. As I told you, in the ventralmost region, there's maximum concentration of dorsal, and this maximum concentration of dorsal activates two genes called twist and snail in just a stripe of nuclei in the ventral midline. And that's the stage where you have transition from syncytial blastoderm to cellular blastoderm. Cells start becoming individualized. They start having their own cell membranes. You can see nuclei are becoming individualized. And this strip becomes mesoderm. Now, twist and snail, they both are required for mesoderm formation. So as soon as dorsal moves in, activates high concentration of dorsal, activates twist and snail, and that gives, or that determines fate of mesoderm for this strip of cells. That also coincides with the onset of gastrulation, where you remember this mesodermal cells, they start invaginating like this. Okay, all these events are happening at the same time. However, in this region, which is neuroectoderm, where concentration of dorsal is low, which means twist and snail are not present here, because twist and snail are only active when you have high concentration of dorsal. So here, with low concentration of dorsal, another gene gets activated, which is called rhomboid in the neuroectoderm. And it's not expressed here because twist and snail, this suppresses rhomboid here in the future mesodermal cells. Now, this was so far for the ventral pattern. What about the dorsal pattern? How dorsal pattern is established? Because in the long run, we clearly see very distinct pattern on the dorsal side of the fly as compared to the ventral side, which means pattern formation should be taking place early on as well. So on the dorsal side, we have a different set of genes called DPP, which is also known as decapentaplegic. It's a TGF beta homolog in flies. Then there are other proteins called tolloid, zirconal, and they all, DPP, tolloid, zirconal, since they are involved in dorsal pattern formation, they are all repressed by dorsal here in this region. Dorsal ensures that none of DPP, toloid, zircnoid are expressed from ventral most region to neuroectodermal region. Zircnoid, which is another important protein together with DPP, is very high, and it specifies amnioserosa. Amnioserosa is extraembryonic tissue here. So this region, the dorsal most region, is called amnioserosa, and zircnoid is involved in specifying its weight. We are soon going to see how DPP actually plays a role in dorsal ventral pattern formation. Now, what we have learned so far, we have, with the activation of toll signaling at syncytial blastoderm stage, dorsal moved into the nuclei in the ventral cells, ventral nuclei. You can see individual cellular membrane is becoming. Now, if you have mutation in, let's say we talked about delta cactus, it means dorsal will move everywhere into the nuclei. We call such a phenotype ventralized. There are other mutants as well, not just cactus. There are other mutants as well, in which dorsal may move into the nuclei all over dorsal ventral axis, and we call such embryo ventralized. Similarly, contrary to ventralized, we have phenotype which is dorsalized, and I already explained you. In case of dorsalized, the dorsal protein is excluded from all the nuclei. It's not within the nuclei, it will be excluded. So this is the wild type. So if dorsal protein is kicked out of the nuclei, it never gets access within the nucleus, just mimicking the dorsal axis. So this phenotype is dorsalized. Now, what we have learned over last 3 or 4 decades is that there is a threshold effect. And by threshold effect means if you push the protein concentration of dorsal beyond a threshold, you will disturb the dorsal ventral axis formation or pattern formation. If you pay attention here in this figure, so again, this is the dorsal axis, ventral axis along the x-axis, and along the y-axis, we have the concentration of dorsal protein into the nucleus. This is showing wild type. So up to this concentration, this maximum concentration, you have, the ventral most region. And then as you can still see, the lower concentration, this can be the neuroectodermal cells, and then dorsal ectoderm, etc. And this is telling you, when you compare this graph with this one, this tells you when you disturb the concentration gradient, or if you go beyond a threshold of dorsal protein, and here you have passed that threshold. So you have increased the dorsal protein concentration in such a way that there's more dorsal protein present. And the threshold is now, which is this one. This is lying here. When you have more of dorsal beyond this threshold throughout, from here all the way to there, you can see dorsal moved into the nucleus. And of course, there's no more dorsal axis. Everything is ventralized. So This is because of, when you have a concentration gradient, dorsal is a transcription factor. Okay. There are, within the genome, there are high affinity binding sites for dorsal. And then there are sites, let's say within the genome, which are low affinity binding sites. When you will have low concentration of gradient, these sites will not be bound by dorsal because there's a very little dorsal protein present. And dorsal will be present only on the high affinity binding sites. Now, when you move the protein concentration beyond a certain threshold, now even these low affinity binding sites will also be bound by excessive dorsal protein present, and the genes which are next to these low affinity binding sites will also be active, and you will have throughout, starting from ventral all the way to dorsal, you will have twist and snail being expressed, for example, and twist and snail will repress rhomboid, and whole uh, dorsal ventral axis is disturbed. Is it clear so far?
Yes, sir.
Okay. So this inhibitory interaction we have already covered, that dorsal, high concentration of dorsal activates snail and twist, and they inhibit rhomboid. Low concentration of, so this is for high, and if you have low concentration of dorsal, you have activation of rhomboid, and then that leads to neuroectoderm fate. And what we have just seen, that just a few fold difference in the dorsal, it determines whether an unspecific embryonic cells form mesoectoderm or neuroectoderm. So the whole fate of cells can be changed due to, few fold difference in the concentration of dorsal protein. It's that crucial. And if you have five-fold difference, the future ventral midline or neuroectoderm, everything is totally collapsed. So what is actually neuroectoderm? Neuroectoderm is, we have talked about neuroectoderm. So here, this is the region, this mustard one, this one. This is the region which is being referred to as neuroectoderm. Now, this is very important because this is going to make the future, some of the cells from this region is going to make the future nerve cord or the nervous system. So there are three layers along the dorsal ventral axis, which form three distinct columns. And these three distinct columns, they are going to make future nerve cord. Cells from this region, they are going to move inwards. They're going to leave their place, and they, as a group of few cells in three different layers, they make neuroblast. Neuroblast are the early neuronal cells or nervous system cells. Now, let's talk about the dorsal. Yeah, Abdullah?
Sir, so the binding sites for rhomboid, they will be of high affinity for dorsal, and snail key will be of low affinity.
Good question. So you are saying dorsal has the binding site for dorsal, and twist and snail are low affinity because only at high concentration dorsal is activating twist and snail. Yeah, according to the explanation for high affinity binding site and low affinity binding sites, your assumption is right. You're thinking in the right direction. But experimentally, if it has been proven, I must admit my lack of knowledge. I can't recall a research paper where they have shown this, but yes, you are right. Should I move on?
Yes, sir.
So now we are going to learn about the dorsal axis formation. This is the dorsal, okay, this region. And the proteins involved are DPP and associated protein. I told you DPP is a homolog of TGF beta cytokine. It should be cytokine. It's a BMP4 homolog. DPP is a morphogen, okay? And there is a concentration gradient. And just like anterior posterior axis formation, dorsal ventral axis formation is also specified by different proteins which are deposited by mother, or what we know as maternal effect genes. If we have absence of dorsal, we know we become dorsalized. And if we have absence of DPP, we'll have dorsalized, because DPP is going to, if we have absence of DPP, we'll have ventralized, because DPP and dorsal, they kind of antagonize each other. They kind of, they have an effect which counter each other's effect. DPP plays a role all the way up to, from dorsalmost region all the way up to neuroectoderm, and dorsal plays a role from ventralmost region to neuroectoderm. So far we have learned that dorsal, it moves into the nuclei, cellularization takes place, and once you have cellularization, cells become individual. Now cells are not having luxury of sharing the freely diffusing transcription factors in the cytoplasm. DPP is a cell membrane or a cell surface protein. After cellularization, most of the time, you have secreted transmembrane proteins and receptors, and they are the one which then transmit developmental signals by, through cell membrane, because now cells are individualized. As I told you, DPP specifies the dorsal ectoderm, and most dorsal region is called immunoserosa. It has the highest concentration of DPP. So if we draw like this is the ventral side, this is the dorsal side, and from here to here, you have neuroectoderm, you have dorsal ectoderm, and then this region is called immunoserosa. And the highest concentration of dorsal, so if we try to see the concentration of dorsal, concentration of DPP, it goes like this, and then like this, a peak and like this. So I try to draw like this. So this is the concentration of DPP. So least to maximum. So this is the maximum concentration of DPP. And DPP is absent all over here because dorsal is antagonizing DPP. If you bring in the messenger RNA of DPP at early stage embryo, the ventral ectoderm, it can become dorsal ectoderm. If you inject DPP messenger RNA, for example, here in the neuroectodermal region or ventral region, everything will become dorsalized. Now, how DPP, so this is the concentration gradient of DPP. You can see maximum concentration of DPP is here. How DPP gets restricted to the dorsalmost region. How such a threshold effect is established. DPP initially is uniform, if you pay attention here. So it's uniform throughout here, but this is slowly restricted. This is due to action of dorsal and other proteins involved, which I'm going to explain you in a moment. So as I said, so DPP is expressed uniformly in the initial few hours, but then within less than an hour, its activity is restricted only to 5 to 7 cell strip, which is this one, which is known as the immunoserosa, and it's much low in the dorsal ectoderm. So initially it was same, but then look, very little amount of DPP in the dorsal ectoderm. There are proteins called SOG, short gastrulation, and tolloid. They modify the DPP activity by interacting with different forms of its receptors and DPP itself. For example, short gastrulation, then twisted gastrulation, tolloid is a metalloprotease. SOG is expressed throughout the neuroectoderm. It binds DPP and prevents it spreading into the ventral region. So what is happening now? So you have SOG, short gastrulation. It's expressed throughout the neuroectoderm, and it will bind DPP. So DPP, which was here, SOG protein is in the neuroectoderm. So it is going to restrict DPP beyond the neuroectoderm. However, tolloid is a protein which is uniformly expressed in the dorsal ectoderm and in the dorsal region. Tolloid, it degrades short gastrulation, SOG, or in all this region, and helps DPP spread or stay in the dorsal ectoderm. Now, as I told, so this is a summary. So short gastrulation, high in neuroectoderm, and then DPP is restricted only in the dorsal region. So short gastrulation is low in dorsal ectoderm, and there is tolloid, which degrades short gastrulation in the dorsal ectoderm, and DPP concentration, it stays relatively high in the dorsal ectoderm as compared to neuroectoderm. In addition, DPP also makes a heterodimer with a protein called SCRU, which is another member of TGF-beta. And then binding of DPP together with SCRU it prevents DPP degradation, and that's how DPP becomes very high in the immunoserosa. Now, what has happened? You have high dorsal, you have high DPP, you have twist snail, which are themselves transcription factor, you have rhomboid, you have SOG here, you have tolide here, and then very high concentration of DPP is established there. DPP plus screw. Cells are cellularized, okay? Cells have become individualized. And these transcription factors are going to activate genes which are downstream of Toll signaling, or which are going to give the mesodermal fade. Here, when rhomboid is there and other genes which respond to low concentration of dorsal, the fate of these cells is established differently. As I told you, they are going to contribute to neuroblast cells, etc. These mesodermal cells are going to invaginate because this is soon going to be followed by gastrulation. And similarly, there will be changes taking place in the dorsal side as well. At very early embryonic stage, all these cells, they look very similar. But if you look at the molecular signature, if you look at the developmental changes which are taking place as a result of these maternal effect genes, these are huge changes, and they are laying down what we famously know as pattern formation. And the man who proposed and who very strongly advocated pattern formation in the early developments as key feature of cell fate determination and specialization of cells is Louis Wolpert, whose book we are reading, and Louis died 2 weeks ago at the age of 91. So such are the people who are really who leave an impact in this world. So after the break, so let's take 10 minutes break. After the break, we are going to go back to anterior posterior axis formation, and we are going to learn about gap genes. From maternal genes, we move into the gap genes. Okay, so let's take a break. Any questions if you have? So let's take break then.